Hello, everybody. Thank you for your interest. My name is Emmanuel from the Transfusion Center in Epernage in Switzerland, and I work on red cell concentrates. So regarding the transfusion of red cell concentrates, a debate has raised if the donor sex may have an impact on the transfused patient's outcome, but observational studies could not yet provide a clear answer. So here in our study, we aimed as a first approach to evaluate the impact of the donors and the recipient sex on red blood cell properties. So for this purpose, we developed an in vitro transfusion model that you can see here. So on the one hand, we have red cell concentrates, two female and two male ones, and we extracted the red blood cells by centrifugation and washing. And we mixed two red blood cells of the same sex together. And on the other hand, we have a pool of female plasma and a pool of male plasma, and these pools represent the recipient. And we incubated the red blood cells together with plasma in a sex matched and in a sex mismatched manner, and at a hematocrit of only 5%. So the idea was here to have a high volume of plasma, to have sufficient quantity of glucose, and to limit the effect of waste accumulation. And then we incubated the reconstituted blood at standard incubation conditions, and we sampled at different time points, so from 0 to 48 hours. And we split samples in red blood cells and plasma. And so we started this experiment with red cell concentrates at storage day 2, and we followed these bags until storage day 42. So here in table 1, we just listed the standard blood parameters to just validate the quality of used red cell concentrates and to indicate that red blood cells are maintained during in vitro transfusion. So here we continue with parameters measured during in vitro transfusion. So at storage day 2 and storage day 42. So the color of the lines indicates the sex of the red blood cells, and the continuous lines are the sex matches, and the dashed lines are the sex mismatches. And here you have the incubation time in hours. So glucose is maintained, and here the difference is just due to processing, since women have a lower hematocrit, so a higher volume of plasma is mixed with a glucose containing anticoagulant solution. And here we can see that lactate correlates with glucose. Concerning the 2,3-DPG, we can see that the clearly rejuvenation process compared to stored red blood cells, and the rejuvenation process is lower at storage day 42. So all transfused red blood cells may be less efficient in vivo. And ATP is also maintained, and even at a higher level than in the red cell concentrates. And here we can see a difference, and this difference may be related to physiological sex differences, may be related to different oxygen saturation, but this difference is offset due to same storage condition. And interestingly, here this difference is maintained during incubation. And here we can see that hemolysis increased during incubation and also with storage time. And we have a higher hemolysis rate in male red blood cells than female red blood cells. So in conclusion, this transfusion model works well, since the red blood cells are maintained in an active state at 37 degrees up to 48 hours of incubation, and 6 differences were observed for ATP levels and for hemolysis rate. So thank you for watching, and goodbye.